All right, everyone, will be just one more minute. Thank you. Okay. Looks like we're all set. Thank you, everyone. It's about 4:30, so we're ready to begin. Thanks, everyone, for being here on Zoom today for the final of our WiSys Quick Pitch showcases. This May, we are wrapping up the series by featuring students from UW-Platteville, UW-Stevens Point, and UW-Stout. Thank you to all of the campus representatives who've made this event possible, and especially thank you to all the students participating today. Couldn't do this without you, especially. My name is Ray French, and I'm the WiSys Regional Associate serving the Western Wisconsin campuses. Many of our WiSys team are behind the scenes today, making sure this event is happening. And as an organization, we're proud to serve the inventors and innovators on our UW system campuses. We're also excited to be able to bring you events like these as part of our vision for building a culture of innovation for a better future. The goal of the WiSys Quick Pitch competition is to inspire students from all academic disciplines to consider the impact of their research or innovative idea and how their findings could one day benefit the economy and society. Hopefully this helps students put the why behind what they're doing. How it works is that each presenter will describe their project in 3 minutes or less, and each pitch will be evaluated on its overall quality. Specifically for the presenter to articulate their topic in lay terms, discuss the problem or unmet need that is being addressed, and share the importance of their findings, drawing conclusions to how their project may impact the economy or society. Today, we'll be presenting the quick pitches from 3 campuses. We're joined by a 4-person judging panel representing their respective institutions. And so first, I want to thank each of these judges for being here today and for your support of the event. We'll do some introductions. So when I call your name, please turn on your video and give a quick wave. We are joined by Dr. Christina Navarro, Director of Athletics and Recreation and Assistant Chancellor for Sports Administration at UW-Platteville. Thank you. Maya Donahue, Director of the Idea Hub at UW-Platteville. Thank you. And Dr. Maria Alm, Interim Dean for the College of Education, Hospitality, Health, and Human Sciences at UW-Stout. Thank you. And Dr. Thomas Gibson, Chancellor of UW-Stevens Point. Thank you. Now, as has been tradition at these showcases, we've invited the chancellors of the participating institutions to make a recorded welcome, but we're lucky enough to have Dr. Gibson with us live tonight for UW-Stevens Point. So I'd like to turn it over to him for a brief welcome. So thank you, Dr. Gibson, Chancellor, UW-Stevens Point.
Thank you, Ray, and good afternoon, everyone. Welcome to the WiSys Quick Pitch Showcase. Again, my name is Tom Gibson, Chancellor of UW-Stevens Point, and one of your judges this afternoon. I'm pleased to represent UW-Stevens Point as a judge for this competition, but I'm also excited to hear the presentations from UW-Platteville, UW-Stevens Point, as well as UW-Stout. This event is an excellent example of the applied nature of higher education, because the information that you're using, you have learned in the classroom, such as applying critical thinking skills and addressing issues or unmet community needs. This is important work that I'm certain that will likely continue beyond your time as students. Indeed, I expect you will continue to apply these skills that you've learned at your respective institutions in your personal and certainly your professional careers for the rest of your life. I wish you the best in this competition, and I look forward to hearing your pitches.
Thank you, Chancellor. Wonderful. Well, now we can get to the main event. So each student presenter will be introduced and cued to start their pitch. Please turn on your audio and video at that time. The timing starts, wonderful. The timing starts when you start speaking. And after each pitch, we will bring in the judges and they will have the opportunity to ask a question. As we move to the next pitch, the judges can finalize their scores. When the pitches are finished, the judges will finalize their scores for the campuses, confer, and determine the winner. And we'll aim to announce the winners by campus at about 6 o'clock. Remember that the winners from each campus win $300 cash prize and are invited to represent their campus at the Quick Pitch State Final on Wednesday, May 26th. In the meantime, presenters and judges, please make sure your audio and video are off until I call your name. And attendees, thank you for joining us today. Best of luck to everyone. So now I'll introduce the first pitch. First, we have Emma Bryla from Biology at UW-Platteville presenting on freshwater mussels of Southwest Wisconsin. You may begin.
Yes. I have a question for all of you. Which of Wisconsin's natural resources do you think plays an important part in the 2.3 billion fishing industry and the 105 billion agriculture industry? If you guessed water, you would be correct. Since water is so integral for our state for economic and recreational reasons, why wouldn't we want to keep it as clean and healthy as possible? You've probably encountered a lake or a stream where you couldn't see the bottom. So wouldn't it be nice if we just had a huge Brita filter for all these systems? What if I told you I had a solution? They're called freshwater mussels. And I'm not talking about the mussels you flex, but the aquatic invertebrate with the hinged shell. Just imagine my hands are the mussels. They are filter feeders with 2 siphons that suck the water in through their bodies, removing contaminants as they go. Actually, one of our native species can filter up to 10 gallons of water just by itself. So just imagine if we had thousands of these animals across our water systems and not just their cousins, the zebra mussels, which are invasive and outcompete the natural mussels. Hi, my name is Emma Bryla, and I'm a senior biology major at the University of Wisconsin-Platteville, and I would like to make you more aware of these unsung heroes. My research team and I surveyed several Southwest Wisconsin waterways to try to fill in the last puzzle piece of a broader Wisconsin DNR mussel survey done back in 2019. Many mussel biologists and citizen scientists set out to update the records from 2003. They found that mussel populations in the northern part of the state are rebounding. And for example, the Fox River, which was dredged out from contaminants, now has a recovering mussel population. Sadly, the southeastern part of the state didn't follow that same pattern. Those populations actually declined from 2003. They weren't able to hit our part of the state, so that's where we came in to try to fill in that gap. We hypothesized that mussel populations will not be doing well in the southwest portion of Wisconsin. Our preliminary research consisted of traveling to 10 different sites and recorded various physical and chemical data like sediment type, dissolved oxygen, water temperature, and water chemistry. Along with that, we also did timed snorkel surveys to see if we could find any live mussel populations. We did not find any live mussels, which could be because there is a lack of preferred habitat and an excess of nutrients. My research team and I have literally come face to face with how important our waterways are when beef cattle, fishing kayakers, and a mom and her 2 kids joined us during our snorkel adventures. We all need to worry about these critters because they drastically improve the water quality by filtering it through their bodies and, in result, making a healthier habitat for the fish that you catch, the water you swim in, and the livestock that rely on this resource. Thank you.
Thank you, Emma. Judges, please turn on your video. Are there any questions? All right.
I guess I can ask a quick question.
Yeah.
Is there any negative to putting freshwater mussels back into our lakes? Because we hear so many times about other invasive species or other problems that are caused by doing something that we think is going to be positive, but then there are unintended consequences. So just wondering if you came across any of that in your research. Thank you.
Right. Yeah, like I said, their zebra mussels are there because they're just invasive and just multiply like crazy. So if we have that diversity from the native mussels, then when the environment does change and then the conditions change, everything won't be wiped out because we have that diversity of native mussels. So yeah, that's a very good question.
Interesting. Thank you, Emma.
Thanks.
All right. Well, judges, you can turn off your video and we'll go to the next. So our next pitch is from Megan Bunyer from Psychology at UW-Platteville, presenting on event memory and emotion related to COVID-19. And just one sec. Maria, you can turn off your video too. And you may begin when ready.
What do you remember about the day you learned that COVID-19 was declared a global pandemic? I know I remember that it was rainy and that when I visited my college for the last time that year, I was struck by how empty the campus was. But was it really rainy? Did I really visit campus that day? My name is Megan Bunyer, and my research is on event memory, emotion, and sense of control related to COVID-19. Previous research shows that memories surrounding emotionally charged events that are out of our control, such as the terrorist attacks of 9/11, may not be as accurate as we think they are. There are many unanswered questions in the psychological community surrounding event memory, especially in regard to collectively experienced emotionally impactful events like COVID-19. The events surrounding the pandemic brought about feelings of uncontrollability. People scrambled to the stores, which quickly ran out of essential supplies like toilet paper. Understanding the nature of the interplay between memory, emotion, and sense of control in the context of COVID-19 may offer insight into various psychiatric disorders like post-traumatic stress disorder or bipolar disorder, as well as fill in many gaps that exist in today's literature. We asked participants to answer questions like, when did the World Health Organization declare COVID-19 a global pandemic, to measure accuracy of memory? We collected their presently felt emotions by asking, at this moment, how strongly or intensely do you feel happy? Or how strongly or intensely do you feel sad surrounding events related to COVID-19? And we gauged how strongly participants agreed or disagreed with statements like, I have little control over the things that happen to me, to measure sense of control. Our findings indicate that levels of perceived constraint or feelings of being out of control exhibited decreased accuracy of memory, and participants with higher ratings of negative and positive emotion were less accurate in their responses. And we also found that those who were experiencing more negative emotions felt less in control. We often feel in these moments of intense emotion that we remember these events with perfect clarity, but research suggests that our memories are less accurate than others for these types of events. Every time we remember something, we are rebuilding that memory, and that is influenced by our current emotions and the stories that we tell. So when you ask yourself, what do I remember about the day I learned that COVID-19 was declared a global pandemic? You may also want to ask, how do I feel? Or maybe a better question, what story am I telling?
Great, thank you, Megan. All right, judges, you can turn on your video. Any questions from the judges?
I'll say, first of all, this is super fascinating. And I know a lot of the research you talked about with like events like September 11th, people, you'd think they'd remember it perfectly, but it's actually all over the place. So your research is kind of tying in like an emotional score to how well people actually remember these things. Is that correct?
Correct, yep. And we found that people who experienced higher levels of positive and negative emotion were less accurate in their responses.
People that like higher, more positive emotions were less accurate?
No, people who were more emotional, like
okay.
both positive and negative, were less accurate.
Got it. Okay. Interesting.
How was scores impacted for folks who tend to agonize or spend significant amount of their time in the past? How did that impact the emotional score?
We didn't measure like rumination, which I think that's what you're talking about. But I have found in the research that exists already that lots of rumination is related to less like feelings of being less in control, which would then, I mean, considering the results here, that would probably decrease accuracy of memory, theoretically.
Yeah. Wonderful. Thank you.
Mm-hmm.
All right. Thank you.
you.
Thank you, judges. All right. Well, next we have Jean Cruz from Biochemistry at UW-Platteville presenting on virus reduction from surfaces using first contact polymers. All right, Jean, you may begin when ready.
Hello, everyone. My name is Jean Cruz, and I've been researching the effectiveness of first contact at removing viruses from surfaces. So I decided to do this because first contact, which was invented at the University of Wisconsin-Platteville, is an adhesive polymer already known for being able to remove fingerprints, spores, and DNA from surfaces. Living through the COVID-19 pandemic as we are, it seemed like a logical hypothesis that viruses could also be removed from surfaces with this polymer. Our research group works with the Planetary Protection Division at NASA's Jet Propulsion Lab to clean equipment by removing alien contamination from returning spacecraft. The threat from viruses like SARS-CoV-2 falls well within this category as a dangerous threat. Our goal is to determine the effectiveness of the polymer for removing viruses from these surfaces using T4 bacteriophage as a proxy for the SARS-CoV-2 virus. Bacteriophage, or simply phage, are DNA viruses that infect bacteria rather than humans. They have a long history as a model organism in basic science research, and based on previous experiments we've conducted, we know that first contact can effectively remove DNA from surfaces, and since phage are DNA viruses, we feel confident it will work here as well. We are currently developing an adapted procedure based on the one used with bacterial spores to accommodate the differences between bacteria and phage. These procedures are being developed to remove the phage from glass, although we have plans to expand the scope of our research to other typical household materials like stainless steel and porcelain. Protocol development is a meticulous procedure, and our efforts this semester are beginning to show promise. Overall, we've been working diligently on demonstrating first contact's ability to remove phage from surfaces because, as we've seen over the last year, there will always be a need for the removal of infectious agents, and we believe first contact can be our universal solution to this.
Great, thank you, Jean. All right, judges, you can turn on your video. Are there any questions?
Jean, your topic is really fascinating and certainly timely. What would a commercial product look like? Would it be an aerosol, some other material? What are you imagining?
Yeah, so first contact, or at least the ones that we're using, are liquid polymers. So they're applied to a surface as a liquid and then left to dry, which is then able to be removed to remove the viruses.
Thank you.
Yep.
Good, interesting. Thank you. Next, we will move to Isabel Dunn. She's with Water Resources at UW-Stevens Point, presenting on benthic macroinvertebrate community responses to wastewater treatment plant discharges in central Wisconsin. Thank you. You may begin.
Thanks, Greg. So I'm Isabel Dunn, a senior water resources major at UWSP, and water is quite possibly our most precious resource. So it's really critical that we understand how to protect it. Every toilet that you flush today or a load of laundry that you ran produces wastewater that ends up in wastewater treatment plants and is then discharged into receiving water bodies, potentially impacting their water quality through contributing excess nutrients and solids, as well as pathogens and contaminants. For the average person generating 50 gallons of wastewater per day, understanding the impacts of the water leaving these plants is important to better manage the quality of their receiving waters. One way to do this is to look at aquatic macroinvertebrates. Aquatic macroinvertebrates are aquatic insects and other invertebrates that live on the bottom of lakes and rivers. Certain invertebrates can tolerate low water quality, while others cannot. So which ones you find can provide valuable insights on the ecological condition of waterways and often tell us a more comprehensive story about the health of water than chemical analysis alone. Using that idea, I decided to investigate changes in macroinvertebrate community metrics, which are just measures of health based on attributes of the bugs present in the water, in order to assess the water quality associated with these plants. I expected to see a decrease in water quality downstream of the plants reflected by the metrics. My project used a control impact study design where I collected macroinvertebrate samples upstream and downstream of the discharge points of 4 plants similarly sized in central Wisconsin. I also collected water chemistry and habitat data at each site to ensure there were no major confounding factors in the environment that would influence these metrics. Using those data, I ran statistical analyses on a variety of metrics whose responses generally fit my expectations by reflecting a decrease in water quality, although overall I did not see a statistically significant change upstream and downstream, counter to my hypothesis. And while future study may include expanding sample size to potentially produce more statistically significant findings, I was able to suggest that the wastewater treatment plants I sampled appear to be managing their effluents well. This isn't too surprising though, as Wisconsin and the United States have rather strict policies and regulations on water quality compared to the rest of the world. Globally, 80% of wastewater flows back into an ecosystem untreated, which contributes to contamination in drinking water that kills 500,000 children under 5 years old each year. So clearly access to wastewater services is a global concern. And I think this approach could be important internationally because it's a cheaper way of determining if discharge from a certain source is affecting the environment. As someone who spends a lot of time collecting and identifying invertebrates, I can tell you that it's relatively cost effective. You can get away with simple materials like a net, some tweezers, and a bottle of preservative compared to the thousands of dollars you'd have to invest in analytical equipment for advanced water chemistry. So all in all, aquatic macroinvertebrates are an important tool for identifying water quality issues and directing funding and efforts in water quality management, both for aquatic ecosystems and for public health. So thanks for listening.
Thank you, Isabel. Are there any questions from the judges?
I guess similar one to what Maria asked earlier. Are there any kind of side effects to introducing invertebrates into some of these waterways?
So most of these invertebrates are already present in the streams. None of this is being introduced, but just by the wildlife that already lives in the streams, we're able to get a signal on the health of that system. So there's all sorts of bugs already present in the streams and lakes around you. So that's what I was looking at mostly.
So just learning from what's already there.
Yeah.
Okay, cool.
Hi, Isabel. I have a question for you. You shared that wastewater could also re-enter our freshwater ecosystem. Do you have a sense of its implication for well water that's not as closely monitored or regulated, if you will?
Yeah, well, all of our water is connected. So anything that we're releasing back into the environment could end up in the groundwater that we drink, especially in the region that we're in, the central sands with super sandy soils. It's a lot easier for contaminants to make their way into the groundwater. So it definitely is a concern depending on the geology of where you are, but especially around here. So.
Okay, thank you.
Mm-hmm.
Great. Thank you, Isabel. Thank you, judges. Okay. Well, next we'll move on to Timothy Lamoureux from Paper Science and Chemical Engineering at UW-Stevens Point, presenting on graphene-based materials for wastewater treatment. Thank you, Tim. You may begin.
So with the implementation of nanofiltration membranes within the U.S. pulp and paper industry, that a potential exists for being able to save energy to the magnitude of around 1.7 million average-sized U.S. homes with the energy saved could have power provided annually. And this energy savings equates to a 30% reduction in the current power usage of wastewater treatment facilities for the U.S. pulp and paper industry as of last year. And so to reduce energy usage by 30% using nanofiltration membranes, a material must be identified that I, Tim Lamoureux, mentored under Dr. Saeed Amir Fakhari, have determined. And this material, it must have some important properties, such as a large surface area, and it needs to let water flow through it easily, as well as it needs to have the ability to control contaminants at realistic concentrations without degrading. And this material is graphene oxide. And so currently graphene oxide is being and has been tested at a lab scale for its effectiveness and efficiency. However, one of the main issues yet to be addressed is taking this technology and scaling it to industry standards. And so this will take some creative engineering to bring this idea to scale. However, at UWSP, we are uniquely positioned with pilot and lab scale technology to encourage the development of long-lasting nanofiltration membranes. And so when developed at scale, this technology would benefit consumers in two main ways. So those of us who are pulp and paper product consumers would see reduced capital costs, which would ultimately benefit the consumers through reduced product costs, hopefully. And there would be a multi-layered environmental remediation effect. With the reduced energy demand, that would reduce the amount of non-renewable energy sources we use, as well as these graphene oxide nanofiltration membranes have the potential to improve wastewater standards by filtering a wide range of organic, inorganic, and biological contaminants. With funding, the next steps we would take would be to acquire equipment needed to develop or synthesize graphene oxide, so we could begin the process of developing these nanofiltration membranes and testing them on a lab and pilot scale to collect industry convincing data, which could lead to further industry consultations. Thank you for your time. I would love to answer any questions.
Great, thanks, Tim. All right, are there any questions from the judges today?
I can ask a quick question, Tim. That was very interesting, but how do you know when you're starting on a study like this, how feasible scaling something like this is? I'm just curious how you gauge feasibility of scalability. Thank you.
So that is a really good question, and it's challenging because there are certain properties that exist for this graphene oxide, which can be controlled. It's just efficiently using those properties manipulated at scale is what is difficult. However, through trial and error, really, I believe, is what can address that.
Thank you.
Yeah, that's great. Okay, thanks everybody. If there are no other questions, we'll move on. Thanks, Tim. So before the next pitch, as I mentioned earlier, we have been sharing welcomes from the chancellors from the participating campuses. And so now we'd like to share these words of congratulations from Chancellor Shields at UW-Platteville.
Good evening. This is Chancellor Shields from the University of Wisconsin-Platteville. Congratulations to all the students participating. You make us proud every day by your dedication and your hard work. I commend you for adjusting during these times and taking advantage of opportunities like Quick Pitch. I wish everyone the best of luck.
Indeed, thank you, Chancellor. Okay, So next we'll move on to Alyssa Marroquin from Political Science at UW-Platteville, presenting on COVID-19 influence on immigration attitudes. Do different frames produce more or less tolerance towards undocumented immigrants? You may begin.
Hi, my name is Alyssa Marroquin, and I'm a political science student at the University of Wisconsin-Platteville. Let me ask you a quick question. Were you aware that we can change the way people think about a topic based on the information that we present about that topic? That is an idea that's at the center of my research. As COVID-19 has rocked the whole of America, undocumented immigrants and non-white citizens have taken the blunt of the physical, emotional, and economic ramifications that COVID-19 has caused. My research looks specifically at COVID-19 and how it has impacted public opinion attitudes towards undocumented immigrants. My project extends the research on COVID-19 and its impacts on the United States. My goal is pretty simple. I want to understand how we can present information about COVID-19 in different ways to alter people's attitudes towards undocumented immigrants. In political science, we call this process framing, or the process of reorienting the way people think based on the information we present. My research presents the pandemic in 3 different frames and then asks survey questions to determine whether each frame did or did not have an effect on people's attitudes about immigrants and immigration. The first frame I use is called the herd immunity frame, and it presents COVID-19 through the necessity of mass immunization, including the immunization of undocumented immigrants. My second frame is called the compassion frame, and it looks at COVID-19 through a little bit more of a moral standpoint, and basically kind of gets at the idea that the COVID-19 virus does not discriminate against who it infects, so we should not discriminate against who we give our vaccines to, including undocumented immigrants. And then my last frame is the economic frame, which looks at the ties between economy and immigration and suggests that immigrants are important to the US's economy, especially when it comes to the agricultural sector. And that should be a motivation for allowing undocumented immigrants to be allowed to have the COVID-19 vaccine. In my survey experiment to test these attitudes on immigration, one of these 3 frames is going to be randomly assigned to a respondent, and then the respondent is going to answer questions about their attitudes towards immigration and immigrants themselves. I anticipate finding that there are going to be significant results on how these frames affect people's attitudes, especially when you look at it through a partisan lens or whether the respondent was Republican or Democratic. I've already conducted a small survey to ensure that the frames I'm using are trustworthy, informative, realistic, and accurate. And this pre-survey has suggested that I'm going to yield significant results when we field the actual experiment. My research is timely and novel. My study is going to demonstrate that the way information is presented doesn't only affect people's attitudes, but people's attitudes towards whole communities that are living in the United States. As the daughter of an immigrant, this information and research is not only important, but it's personal to me. My research is going to shed light on how to best communicate information about COVID-19 and how this information will directly affect people's opinions towards undocumented immigrants. Thank you.
Great. Thank you, Alyssa. All right, judges, do we have any questions?
I always have at least one question. Hi, Alyssa. Have you posited which frame would produce the greatest positive attitude towards immigration?
I have hypothesized about it. Yeah, I haven't gotten any concrete results for, based on the research I have kind of accumulated, it's suggested that the compassion frame is going to garner a lot of support specifically among that Democratic partisanship, but it also suggests that the economic frame might have greater results with that Republican partisanship. So it's kind of between those 2 at this point in time.
Got it. Thank you.
So I want to say you used one jargon word, but you explained what it was, and that was framing. So nicely done. A lot of these pitches just throw out a lot of jargon, and people are lost when you put it all together. So that was really well done. Tell me a little bit more about how you expect this information to really have an impact. You talked about kind of like, it sounds like kind of like an education standpoint, like how we present information to the public. Could you just go a little further on that?
Yeah, so I previously did some research about just general framing on immigrants and how it's affected people's attitudes toward them. And a lot of that research looks at how information in the news is presented a lot of times through negative frames and how that has really affected the United States community's attitude towards immigrants in a negative way. And I know personally, like my father has told me stories about how his attitudes towards him were affected by what people have heard. So what I'm hoping to find with this is ways to communicate the information about immigrants and COVID-19 in a compatible way, but also in a beneficial compatible way so that they're receiving these vaccinations at the same rate US citizens are, because in the end, that's going to be beneficial for everyone.
That's great, Alyssa. Thank you very much. Thanks, judges. Okay, moving on. Next, we have Eric Norton and Travis Newham from Food Science and Technology at UW Stout, presenting on water test kits, coliform, and E. coli detection. You may begin.
Well, hello and welcome. I'm Travis Newham.
And I'm Eric Norton, and we are both food science students at the University of Wisconsin Stout. And today we're here to talk about E. coli contamination of in-home drinking water and other municipal and city water sources. So first off, what exactly is E. coli? E. coli is a type of coliform bacteria which originates in the digestive system of mammals and mainly humans. And they're known to cause illness when ingested by releasing toxins that cause vomiting, cramps, and nausea.
And according to a research article that we discovered titled Emerging Infectious Diseases, each year there's about 73,000 illnesses related to E. coli infections. Many of these are attributed to water contamination. Commonly, E. coli illnesses contracted in the home go undiagnosed and unreported. It is difficult to find cheap and effective test kits that also show a level of precision that does not require specified lab equipment or knowledge of lab techniques so that the average person can test their own water.
So we have been performing tests in our own microbiology lab to prove the level of sensitivity in our tests and also kind of examine the general characteristics of coliform growth so that we can develop a water test kit in the form of a capsule that just exploits the properties of E. coli and coliform bacteria to create a simple color change that's apparent after 48 hours. This kit is, it uses 3 easy to use materials, which is just 1 protein enriched water, a scientific grade plastic bag, and the special E. coli detection capsule that we have developed.
These tests are easy to use and be incubated at room temperature, unlike other commercially sold kits, lending their use to a general consumer use at home. These kits will make it easy and affordable, likely around 50 cents per kit for people to test their own water for contamination and save themselves from needing costly medical attention. We believe that these test kits will revolutionize how consumers at home and companies abroad will be able to verify the safety of the drinking water and other contact water sources from the perpetual E. coli threat. We would like to thank you for your time and interest in our cause. May you all have a wonderful day.
Great, thank you both. All right, judges, you can turn on your video and ask any questions you may have.
So this is cool stuff. One of my questions is, what prompts somebody to know that they would need an E. coli test? Like, if some, how would you even know that like you should get the test to begin with? Is one thing I'm kind of wondering about.
Oftentimes, especially in older cities, the city water infrastructure is very old and crumbling. And otherwise, if you don't live in an older city, oftentimes you also live in the country, and you have some more like well water. E. coli can get into well water very well, just as well. If you find that sometimes you drink your water and you don't feel so good the next day, or maybe someone visits your home and they're not used to your well water and they get sick from it, that's perfect time that you should probably be testing your water.
Travis, it seems to me that there should be a number of products in this space. Who are some of the competitors that are in this space, and how do you think you can deliver a product at a lower price point in comparison?
Yes, there are many competitors. Yes. The thing is, as we've done lots of research on this already, the majority of them want you to send samples to a lab, one of their own labs, which costs a whole bunch of extra money and time. That's one of the things that this specific test kit that we're trying to develop is trying to avoid. It'll only cost the price of the materials, and that's it. It can be done at home, specifically at room temperature with no fancy equipment. You just got to have our correct size bag and our specified E. coli testing capsule that we are working on.
Perfect, thank you.
Very good, gentlemen. Thank you. All right. So next we have Bethany Slimbarski from Sustainable Energy at UW-Stevens Point presenting on Woody Biomass Energy Outreach Program to address Wisconsin Public School District's K-12 barriers and challenges to using woody biomass energy. All right, you can begin when ready.
Thank you. As mentioned, my name is Bethany Slimbarski. And what if I were to tell you that there's a possible way for Wisconsin public school districts to lower their energy costs, gain an on-site teaching tool, and support their local economy to a greater extent? How, you might ask? Well, in the consideration of using a woody biomass energy system. With Wisconsin being comprised of so much forest land and forest product industries, there's naturally this excess production of woody biomass residue or woody biomass byproducts that are currently being wasted. They're left on forest floors or sent to the landfill because they don't know what else to do with it. So school districts seem to show a potential solution to this excess residue problem, and that a lot of them are located in more urban areas where urban wood waste is a major problem, as well as over half of school districts having their own school forests that they could be removing this excess residue from. It's known of a handful of school districts in Wisconsin that currently use wood energy actually quite successfully. And so this shows that it is possible, but it also raises the question as to why. Why are there not more school districts using wood energy? And so that's where this study comes into play. We were interested in assessing school districts' knowledge and familiarity with woody biomass energy, as well as their perceived major barriers to implementing such a system. And also we're interested in their preferred ways of receiving this energy information. And so results from our findings showed that, results from our study showed that there's overall this low familiarity and knowledge of woody biomass energy, and major barriers identified included building infrastructure costs, the additional maintenance required, problems trying to access quality fuel, as well as dealing with the politics of the utility companies, engineers, and the school district itself. It was also shown that many of these districts are going to the Focus on Energy program to receive their information and prefer to receive this information in the form of in-person experiences, online resources, or written reports. And so with these findings, it can help us guide future outreach efforts to these school districts, which could take the form of partnering with or modeling after Focus on Energy, presenting information through in-person experiences, online resources, or reports in order to bring this option of wood energy to the table of these school districts, particularly bringing to the attention the opportunity of funding resources to reduce those costs, as well as helping them make those connections to quality fuel. And then with this added information and knowledge, hopefully generate enough interest to overcome those political barriers. It's suggested that future research look into the impact of this suggested outreach, but if it is successful, it could help generate enough interest to encourage the development of a woody biomass market which would then not only economically benefit these school districts, but also their surrounding communities. Thank you, and I'm happy to take any questions.
Great, thank you, Bethany. All right, judges. Do we have any questions?
Bethany, that was super interesting. Thank you very much. If I can just ask a question, did the schools or anyone raise any environmental concerns? Because I'm guessing you're burning this biomass, and I just know that some cities have had issues regarding this. So I'm just wondering, was that part of a cost barrier as well? Or how did you address that issue? Thank you.
No, that's a good question. And we thought that too. We thought environmental issues would be a really big barrier for a lot of these schools. So we did include it in, we sent out online surveys, and like nobody selected that as being a problem. I think because of the new systems coming out, they burn very efficiently, especially these gasification systems. And so it's not really a major concern to them anymore. It would be more so for the older systems because they weren't as efficient in their burning. So yeah, it was interesting. Wasn't expecting that.
Yeah, interesting. Thank you, Bethany. Thanks, judges. So next we'll invite up Caitlin Timmons from biology at UW-Platteville, presenting on preventing and preserving microbial contaminants from space. Thank you. You may begin.
NASA's Planetary Protection Division is tasked with protecting Earth and other bodies from contamination by foreign life. However, we can also contaminate space with microbes from Earth. Perseverance is currently looking for signs of ancient microbial life on Mars. Imagine what could happen if it found living bacteria, but due to improper handling, we were to destroy the first signs of life in space by exposing them to bacteria from our planet. My name is Caitlin Timmons, and I am a senior studying biology at UW-Platteville. I am researching the abilities to remove and isolate bacterial spores from different surfaces. This was a task granted to my research advisor, Dr. Mark Levenstein, in collaboration with NASA to develop improved cleaning techniques of spacecraft. Per recommendation of NASA, I use the spore-forming bacteria Bacillus atrophius, which presents a low risk of human infection. Some bacteria form spores to protect themselves from extreme conditions, such as severe temperature ranges, high humidity, and even the vacuum of space. When in the spore state, the bacteria is dormant and cannot be killed. Bacillus species are highly hydrophobic, meaning they don't mix well with water and can adhere tightly to surfaces, making removal an extreme task. So if they are this difficult, how can we remove them? First Contact is an adhesive polymer invented by UW-Platteville professor Dr. James Hamilton at Photonic Cleaning Technologies. Think of it like liquid tape. While it is applied as a liquid, after it dries, it can be peeled off with a lower adhesion than Scotch tape. My research goal is to develop methods that capture and isolate microbes by taking advantage of the unique adhesive properties of First Contact. I have been able to show that First Contact can remove bacterial spores from different surfaces, and those spores can then be isolated from the dried First Contact by dissolving it back into a liquid. This will be effective in future space exploration to reduce forward and backward contamination. So, if spores can survive in space and are difficult to remove, what other problems could come out of this? Scientists can and have fooled themselves into thinking something came from space when it was actually from Earth in the first place. This is known as false positive results. While it is important to clean spacecraft materials before liftoff, it is even more vital to clean them upon return. While I mentioned we don't want Perseverance to destroy what we find on Mars due to contamination from Earth, we would also not want to be on the reverse end of that and experience a bacterial alien invasion. Using First Contact to clean spacecraft upon return, we can not only remove the bacteria from the surface, but also isolate them to discover life from space. Thank you.
Great, thank you, Caitlin. All right, judges, any questions?
So just kind of a clarification. One thing that I'm really interested in is, with science and progress, there's this concept of the precautionary principle, and before we just lunge right into something, let's know what we're doing. Let's look at, a classic example is DDT, which, we're spraying at all of our kids at picnics and whatnot and turned out to be really terrible. Is that kind of what you're going for here? It sounds like we don't know what could happen, but we don't want to have something really terrible happen because we weren't being very careful. Is that, just to kind of clarify, is that sort of what you're looking at?
Yeah, so Dr. Hamilton reached out to the biology department so that way they could properly say if it can remove bacteria then. That was also something NASA asked. But yeah, there was a case back years ago where they thought they found bacteria on the moon, and it turns out it was just from the telescope that they had sent to the moon, and that's how we found out that they can survive in space, and kind of fooled them. They got really excited, so that's where the false positive results come from. But yeah, it's just that preventative measure, but then also it is a way that while we clean it upon return then, and then we can isolate those, that's how a good way that we would be able to find bacteria from space now.
Hi, Caitlin. I have a question. I guess more for clarity. So does the first contact polymer eliminate the bacteria, or does it change the physical property and one still needs to discard it at a later point?
Yeah, that's a good question. So unlike other cleaning measures that kill bacteria, first contact actually only removes it. So when you remove the first contact that contains the bacteria, if I don't go through with the process of isolating it, it does need to be disposed properly in the proper disposal bins then for that.
Okay.
Interesting. Thank you, Caitlin.
Thank you.
All right. Next up, we have Emily Eberflus from Biology at UW-Platteville, presenting on differentiation of mouse myoblasts. Thank you. You may begin.
Hi, my name is Emily Eberflus, and my research this semester surrounds understanding the ins and outs of stem cell differentiation. When you hear stem cells mentioned in the news and other media sources, you may think of real-life clones or mad scientists in a lab growing body parts in test tubes. As dramatic as that may sound, stem cell research has a much broader application in countless areas of science. As a pre-med student, my specific interest lies in how stem cells will impact the future of medicine. Because human embryonic stem cells were first isolated at the turn of the 21st century, stem cell research is a relatively new field with a lot of potential to contribute knowledge about things that no one has thought of or discovered yet. Currently, my focus is gaining experience with certain stem cell related techniques, like determining their preferred growing conditions, learning what events impact their fate, and how to manipulate them to fit specific goals. Currently, I am working with mouse-derived myoblasts that eventually differentiate or turn into myotubes. These are the cells that make up skeletal muscle. I've had success growing and differentiating these cells using a growth media called DMEM, supplemented with 10% fetal bovine serum. They are fed twice a week and passaged once a week, which keeps them from becoming overgrown. This is one of the most important factors in getting the cells to differentiate. Through cell counting trials, I was able to determine how quickly the cells grew and how the growth rate was affected by the number of cells originally seated in the flask. On average, the cells can take up to 39.4 hours to double the population size. I intend to apply all of this information to the study of human embryonic stem cells. These cells are pluripotent, meaning they can differentiate into all three of the cellular lineages: endoderm, ectoderm, and mesoderm. Another way of saying this is that they can differentiate into all three, or they can differentiate into any type of body cell. This will eventually give medical professionals the opportunity to fully control their differentiation, which could have endless applications in the field of regenerative medicine. One day, this may lead to doctors being able to grow hearts from just a few cells or perform organ transplants with minimal chance of rejection because the organ was grown from the donor's own tissue. Unlike the cheesy science fiction plot I described at the beginning of this pitch, growing organs for transplant is a novel endeavor and a technical tour de force. In the end, this will prove to not only be a great scientific advancement, but also a chance to save tons of lives. Thank you so much for listening, and I hope that I've convinced you of the importance of continuing research in this field.
Great, thank you, Emily. Okay, judges, do you have any questions on this pitch?
I don't have a question, but just a comment. This is really, really fascinating. And you have me kind of imagining about what all of the future applications could be for this new technology and research. So it's really interesting.
I'll echo that. Just because nobody has questions doesn't mean we don't care. We weren't paying attention. I think we're all like, wow, which is really cool. So yeah, good stuff.
Thank you.
Yep, thank you. Thanks, judges. It's funny to, as the judges turn on their cameras, sometimes you see them. Oh, interesting. So I appreciate that, and absolutely. And just to echo, all of the pitches are doing a phenomenal job giving us interesting topics to think about. This is great. So thanks, everybody. We'll move on next to Jacob Weller from Criminal Justice at UW-Platteville, presenting on Hey Bud, Let's Not Party, an evaluation of participant engagement in written assignments within OWI and drug treatment courts. That's been my favorite title so far. Thank you.
Thank you, Ray.
All right, Jacob, you can begin.
Perfect. So the American Addiction Centers released a statistic stating that 60% of Americans who receive substance abuse treatment relapse after this treatment. That number is far too high. And that's why I, Jacob Weller, conducted a study in an attempt to combat this. So I focused on a single court within the criminal justice system. It's called an alternative treatment court. Alternative treatment courts are an alternative to the standard prison, jail, and probation model in which participants receive substance abuse treatment or counseling and serve their sentence. So within this treatment court, I looked at two specific factors: participant engagement through written assignments distributed by the court and reoffending rates by the participants. So let's start with the participant engagement through written assignments. So I was able to gain access to homework assignments practically that were distributed by the treatment court coordinator and to the participants. And these participants would respond to different prompts varying from either goodbye letters to the substance that they were using, or basically just writing about their experience. And I counted up all these sentences that the participants wrote and analyzed them to see who was more engaged compared to their peers, and wanted to understand, like I said, the level of engagement. And the reoffending rates, that's basically a binary thing, where it was yes or no. Did the person reoffend after entry/graduation to the court? Yes or no? And after my analysis of the actual two variables, I found that there was actually a trend towards people that are more engaged or who write more sentences, that they are less likely to reoffend actually in the future compared to someone who does not write as much. They are more likely to reoffend in the future. So these results are very significant because it shows that participants are having some type of actual experience while writing during the treatment court experience. And this could be from either a transfer, because it's transformational, about them reflecting on their usage or their support systems that it affected, or even the community that maybe the crime that they committed affected. Or it could just be that they were sincere and thoughtful in their responses. But regardless of what was a motivator to this, I hope that my research and other research in the field of academia will be able to actually help lower that 60% statistic that I mentioned earlier, because that number is far too high, and we need to make sure that our communities are drug-free in a sense that everyone is having a safe environment to live in. Thank you for listening.
Thank you, Jacob. All right, judges.
So this is goodbye letters that people write to their addiction. Is that correct?
That was just one example. I studied 9 different types of letters that these people actually wrote.
Cool. And shorter letters means more relapse. Longer letters means, is that in that case?
In a sense, yes. There was a trend towards that, but there was a few cases where someone would be very engaged, meaning that they would write a lot, but still reoffend in the future.
I guess I had a very similar question. I wanted to better understand how you were assessing engagement beyond length of the assignment. Were there any other characteristics or criteria that you leveraged?
Not necessarily, because level of engagement, that is that's a very personal level, because some people may not just write as much, or it may be a little more quiet compared to someone else. So actually being able to have distinct and tangible documents or experiences by these participants was very hard to come by. So the only thing that was really readily available was these actual documents. So I could have looked at how many pages a person wrote, but that would not have been very good, because someone could write 2 words on a page versus someone that could write the whole entire page. And in a sense, those two would be deemed as engaged the same level, but that's not the case. Or I could count the number of words, but that would take far too long, and the resources were simply not there to count every single word that these people wrote. So I found that the sentences in describing the levels of engagement was probably the best middle fit for that scenario.
Got it. Thanks, Jacob.
Yep, thank you.
Cool. Thank you. Thanks, Jacob. Thanks, judges. All right, moving on. Next, we have Claire White from Chemistry and Music at UW-Stevens Point, presenting on ester concentrations in beer yeast based on fermentation temperature. Thank you. You may begin.
Hello, my name is Claire, and I am a music and chemistry student at UWSP. Today, I will be talking about one of Wisconsin's most beloved beverages, beer. Oftentimes, when you arrive at another person's house, they'll ask you, "Can I get you something to drink?" But here in the Midwest, we say, "Can I grab you a beer?" Because of the widespread popularity of beer in the Midwest, this allows for the beer market to be very prominent, but also competitive. While this provides excellent production of fantastic beer for us Wisconsinites, how do these breweries compete to produce the tastiest and most unique beer? Although there are many sources on optimizing beer taste, there are close to none on the correlation between ester concentration and fermentation temperature. Many brewers are unaware of this chemical aspect of brewing, and companies do not advertise it. So, in my research, I'm focusing on the varying ester concentration in beer based on their fermentation temperature. Esters are a natural byproduct of the fermentation of yeast and provide different flavors and aromas in the beer. In this project, I brewed a light IPA beer and then fermented it at varying temperatures from 60 to 70 degrees Fahrenheit. I extracted these different esters, such as ethyl hexanoate, ethyl octanoate, and phenyl ethyl acetate, but these names might not mean anything to you at all. But they provide different flavors and aromas, such as apple, banana, pineapple, and more. So I was able to obtain these concentrations as well by analyzing them on the GCMS, which stands for Gas Chromatographer and Mass Spectrometer. And then along with some UWSP professors, I blindly tested some of these different beers and could determine that the ester concentrations that I obtained correlated with the flavors that we were able to taste. So this is a very promising result of the research because it can provide useful brewing and fermenting data to the local brewery that we are collaborating with. So with this information, a brewer can select a yeast fermentation temperature to more predictively achieve a specific ratio of esters and flavor profile. So as we continue to learn more and more about the chemistry of beer making, only the best breweries will be able to craft tasteful beer using different fermentation temperatures along with other techniques. So my research will not only provide this useful information, but it also has the potential to inspire new ways of scientifically customizing beer. And I'm excited to continue this research in the fall and to provide brewers with prevalent results so we can keep Wisconsin doing one of the best things it does, and that is brewing beer. Thank you.
Thanks, Claire. That was great. Any questions?
So Claire, your focus group subjects, they are all of age, I'm assuming, correct?
I'm sorry, can you repeat that?
Your focus group subjects.
Oh, yes.
All age.
Yes. I turned 21 in August, this last August, so I made the cut,
Okay, just joking. So I'm trying to better understand the difference or the variation in temperatures. Do that correlate to a particular taste? Tell me how does the varying or the variance of temperature impact the quality or the taste of beer?
Yeah, so typically the higher the fermentation temperature, the more production of esters. So a lot of esters do produce fruity tastes. So just looking at that, typically if the temperature is higher, there's going to be a more fruity taste. But also getting to certain temperatures will affect different aspects of it. Like it could strain the yeast. So maybe, you have to be careful about fermenting it too high because it can affect other things in the beer.
Thank you.
Yeah.
All right. Thanks, Claire. Just echoing your sentiment of helping Wisconsin do what it does best. So thank you for your work.
Thank you.
All right. Now we'll head to a second little break here. We'd like to share next these words of congratulations from Chancellor Frank at UW Stout.
Good evening. I'm Catherine Frank, Chancellor at UW Stout, and the Quick Pitch competition is aimed at encouraging innovative and entrepreneurial thinking, and this is what we do best. UW Stout students, I know that you are well positioned to excel in the competition. UW Stout commends all of the students participating in the Quick Pitch competition and wishes everyone the best of luck. Go Blue Devils!
Great, thank you, Chancellor. All right, 2 more pitches to go. So next, we have Miter Yang from Food Science at UW Stout, presenting on elevating nutritional and antioxidant capacity of fruit, vegetable, and cranberry pomace infused value-added cookies. All right, you may begin when ready.
All right. Hello, everyone. My name is Miter Yang, and I'm very excited to be here with you all. As mentioned, I am a graduate student at the University of Wisconsin Stout, and my supervisor is Dr. Pranabindu Mitra. Per the 2020 to 2025 Dietary Guidance for Americans, recently published this last December, we know that about 80% of the US population does not meet fruit intake recommendations, and that about 90% of the US population does not meet vegetable intake recommendations. To add to this piece of information, fruits and vegetables are also significantly wasted per year. This is evident in industries such as the fruit processing industry, that produces millions of pounds of fruit pomace, such as cranberry pomace each year. Pomace is the remains of pressed or crushed fruit, usually containing the stem, pulp, and skin of the fruit, which cannot be directly disposed of in the fields due to uncontrolled fermentation. Therefore, an issue that companies are currently facing is having to spend large amounts of money to treat these byproducts before their disposal. However, what if all of this could be solved by converting this type of byproduct all into one cookie? So my research this year focused on elevating the nutrition and antioxidant capacity of fruit, vegetable, and cranberry pomace infused value-added type cookies. Cranberry pomace is a well-known source of bioactive compounds such as polyphenols, as well as vitamins, provitamins, and dietary fiber. So in this study, fruit cookies and vegetable cookies were infused with varying amounts of cranberry pomace. And then they were analyzed for different nutritional parameters such as carbohydrates, lipids, proteins, moisture, ash, fiber, and antioxidant capacity. So our results show that an increase in cranberry pomace addition did significantly increase the fiber content of cookies from about 1 to 5%, and the antioxidant capacity from 3.5 to 12.5 times higher. Vegetable cookies also showed a higher fiber content than fruit cookies. So due to its nutritional benefits, these value-added type cookies do indeed have high potential for commerciality. Furthermore, cookies are already a common baked good item that is familiar and well accepted by consumers. So instead of wasting cranberry pomace and having companies spend more money to dispose of this type of byproduct, cranberry pomace can be converted into an edible food ingredient used in baked goods such as cookies. Not only does this help enhance nutrition, but it also brings in profit for food companies and helps reduce food waste at the same time. To add, these products would help expand and encourage more development of nutritious, sustainable food type products. This expansion not only advances and diversifies the market, in my opinion, but it also helps to make it more competitive and innovative overall. So the next time that you reach for a cookie, you could be gaining fiber and nutritional benefits all in one yummy snack. Thank you so much for your time.
Sounds good to me. Thank you. All right. Any questions from the judges?
I have a question. Miter, I don't know if it's the time of the day or not, but these cookies really seems to be quite interesting to me. I wonder if the cranberry pomace is a proxy for a sweetener or other some type of sugar or glucose.
So we didn't test to see if specifically cranberry pomace contributed to the sweetness or overall sugar content of the cookies. But for sure, when we conducted small sensory evaluations among me and other research students, we did find that the fruit type cookies were definitely a lot sweeter than the vegetable type cookies. And this could be just attributed to the amount of sugar that is in fruits versus in vegetable type ingredients. So yeah, those cookies were definitely a lot sweeter, and I would suspect that it would be quite popular among children versus the vegetable type cookies.
Thank you.
So I just want to say I'm a big fan of the idea of a lot of pitches get very complex. And one thing we're going to be doing at UW-Platteville for some of the events is we're going to actually look to have people pitch to a panel of 10-year-olds to make sure those 10-year-olds could understand. And I'll just say right now, your pitch would win because you said the word cookie like 27 times. And I think they'd just be like, yes to the cookies pitch. So no, really good stuff. This is awesome. In all seriousness, finding ways to make cookies healthier is just a really great thing, because it struck me many times that, isn't it bizarre that broccoli is so healthy for us and Twinkies are not, but yet which one do we want all the time? So, your research kind of looks to sort of shift that around. So very good stuff, liking a lot.
Thank you very much for your time.
Great, thank you. That's great. Thanks, judges. All right. For our last pitch, we have Yushan Yang from chemistry at UW Stevens Point, presenting on synthesis of a novel solar absorber. You may begin.
Hi, everyone. My name is Yushan Yang, a student studying chemistry department at the University of Wisconsin Stevens Point, and I'm from China. Today, I want to talk about my research in progress, which is based on the synthesis of a novel solar absorber. When you hear this topic, you might think, what is a novel solar absorber? What kind of function will it have? How is it related to our lives? Let me tell you. Novel solar absorber is a new material that can absorb sunlight and convert it to electrical energy effectively for our human use, like use in renewable energy technology. You may be wondering, why do we need new solar absorbers? Don't we already have the materials that can absorb sunlight for electricity? While the short answer is yes, we have solar cells that do this, and at the heart of the solar cell is the solar absorbers. But current solar cell technology has its drawbacks and some limitations. The novel solar absorber is made from Earth's abundant elements that can absorb sunlight better than the traditional solar absorbers. This potentially will result in lower cost, more flexible, more lightweight solar cell panels. In my research, I'm trying to synthesize a special class of materials that meet many of the criteria for an ideal solar absorber. The synthesis method involves mixing simple metal salts with sulfur powder in an organic solvent at moderate temperature. By optimizing the reaction conditions, such as temperature, time, mental salt, I hope to discover reaction conditions that can lead to the desired solar absorber. Finding ways to improve and using renewable energy in our everyday lives will make our lives better. We still have some space in solar energy research and find befitting materials to improve its efficiency on electricity. Overall, the solar absorber I'm trying to make represents just only one in the class of novel solar absorber to be discovered. And if you have any question, you can ask me. And thank you for your listening.
Great, thank you. All right, judges. Are there any questions on this pitch? I was wondering, actually, if you could say again the materials that you're looking to use, the more common earth materials that you were looking at using.
Yeah, like the metal tin and some more common metal like around our lives is reduced the cost a lot than the traditional solar cell.
Right, yeah, absolutely. That's a great, that'd be great to use those more readily available metals. Very good.
Thank you.
Thank you. All right. Well, that concludes this evening's pitches. Congratulations to all of you. Thank you. Thank you. Thanks to all the pitchers. Thanks to all the judges. So congratulations to everybody. A virtual round of applause to everybody. So now what we will do is ask the judges to mute their audio and video. There's a conference line that they can join as they finalize their scores for the awards. And so while we do that, we want to share a few words from the campuses represented here tonight. For the previous two showcases, we featured videos from the participating campuses. Some have focused on undergraduate research, like this first one from UW-Platteville, created specifically for tonight.
Undergraduate research allows students to gain a number of different transferable skills. For example, almost in any undergraduate research project, something will go wrong. This gives students the chance to problem solve and troubleshoot, which is a valuable skill that they will need in any career going forward. So in addition to transferable skills, students are oftentimes working very closely with their faculty mentor on research projects. This often opens the door for future opportunities in graduate studies or careers and allows the student to form networking relationships that they can potentially use to further their career in the future.
Students at UW-Platteville can apply for scholarships to support their undergraduate research activities through the Student Research and Engagement Fund, as well as other opportunities such as the Summer Undergraduate Scholars Program, which provides funding for students to do research full-time with a faculty member through the summer. We partner regularly with WiSys on events like the Quick Pitch competition, the Innovation Mixer, and the Prototype Hackathon. And these are events really designed to help students develop the skills necessary to take their innovations and their creative ideas to the next level. Working with outstanding, creative, and approachable faculty here at Platteville who care deeply both about teaching and research, students here truly have the opportunity to become pioneers in whatever field they are in.
There we go. Thank you. Thank you, UW-Platteville. That was great. We love that presentation. And we certainly do appreciate our continued partnership with you as WiSys and all that we're able to work on together. So next, we're going to showcase UW-Stevens Point with a nice video presentation. Thank you.
We are here for a purpose. Some arrive knowing it. Others are still searching for it. But this we share. The determination to create the future we were meant for. motivating and encouraging each other to work harder, reach higher, and be who we are meant to be. We are the University of Wisconsin-Stevens Point, where challenges don't hold us back. They invigorate us, and we will always rise to meet them. For a better future, a better Midwest, and a better world. Discover your purpose.
Great. Thank you, UW-Stevens Point. Now, if that doesn't get you jazzed, I don't know what will. I love that kind of music, and there's a whole thing on motivational videos on YouTube, and that totally just struck me as that. So love it, and love UW-Stevens Point. All right. Finally, we've got one more video from UW-Stout.
Ready. It's a word often used to describe UW-Stout's career-focused graduates who are ready to hit the ground running. Even in this time of uncertainty, Stout is ready too. Ready to provide our students with our applied learning options in person or online. Don't put your future on hold. The world is changing. Be ready to change with it.
I love these musical selections. That's awesome. Thank you, UW-Stout. I'm also a sucker for campus aerial footage. So many campuses do a great job showcasing how beautiful they are. And we have such beautiful campuses all across our state. We are blessed. So thank you to all the campuses who've participated in Quick Pitch. As I mentioned, this is our final of the 3 Quick Pitch showcases. And I think for the first time ever, we've had all 11 campuses that WiSys works with participate in the same year. So that's phenomenal for a pandemic year, and just congratulations to everybody. So now I think we'll pause for just a quick moment while I check in with the judges as they finalize their scores, and we will announce the winners in just a few moments. Thank you. And for those of you just joining us on YouTube, just a quick note, we will be right back with the award winners shortly. Thank you. All right, everybody. With a minute to 6, we are back to announce the winners of the Campus Quick Pitch Showcases. So first, another congratulations to all the students who presented today. You all made great strides in the last few weeks as you prepared for today, and we commend you for taking part in this competition. Also, you're either in finals week or finals are starting next week or very shortly. So kudos again for taking on this competition. And hopefully it's been a great experience for you. So now we'll talk about the awards. The first place winners from all the campuses today win a $300 cash prize. And second place winners will be available at Platteville and Stevens Point, taking home a $100 cash prize. The first place winners today are also invited to represent their campuses at this year's Quick Pitch State Final, which will be on Wednesday, May 26th at 4 p.m. So mark your calendars and join us again on YouTube for that. So as we begin, I'd like to ask all the award winners to stay on after we conclude for a quick screenshot picture of everybody. And students, you should also be prepared that if your name is called, we'd like you to turn on your video and just give us a quick wave. We'll snap a screenshot of the campus winners as well. So now the moment we've all been waiting for. Judges, thank you again for your work, and students, again, for your time and participation here. We will start with announcing the UW-Platteville winners. Starting with second place in the quick pitch competition goes to Alyssa Marroquin. Congratulations. And finally, first place for UW-Platteville's quick pitch competition is Jean Cruz. Hey, thanks, Jean. Congratulations to you both. Alyssa, Jean, could you take a quick second to pop back on? All right, let's give us a smile and we'll do a little screenshot. Thank you. Awesome. Wonderful. Congratulations to the winners from UW-Platteville. Next, we'll move on to UW-Stevens Point winners. Second place goes to Claire White. Yay, thank you. And first place for UW-Stevens Point quick pitch is Isabel Dunn. Great, congratulations. Now would you both turn on your videos again? Awesome, thank you. And perfect, good smile, all right. Thank you both and congratulations. Last but not least, we'll announce the winner for UW Stout. And the winner for quick pitch at UW Stout is Miter Yang. All right, congratulations. All right. Well, thank you, everybody. Thank you for sticking with us tonight. This concludes our WiSys Quick Pitch May Showcase. Congratulations to everybody who participated and also to today's campus winners. Thank you to our panels of judges. You've been a great help in helping us put this together. Thank you to our campus research partners who supported all this activity, and especially to the mentors who guided students through this program and all the training that goes into putting something like this together. And thank you to all my colleagues at WiSys for your parts in making this event possible. And thank you to all the attendees who supported these students and all the campuses participating today. Join us right here on May 26th for the WiSys Quick Pitch State Final at 4 p.m. So lastly, thank you and have a great night.